Welcome to the last session of the afternoon. What are the later life contributions to reserve, resilience, and compensation? I think this is an historic symposium. It's the first time in my life that I have ever chaired a symposium or been on one that has all women on it. Now you may notice Bob here. He's obviously not a woman, but he's our cameo speaker, but we're very excited to have just a set of excellent presentations today. So our first speaker is Sara Burke, University of Florida, an animal model of cognitive aging. Do compensatory neural processes confer resilience?
Thank you. It's an honor for me to be here. I would like to thank the organizers and also Denise Park, our session chair. I'm really proud to be kind of the rat representative on this panel. And there's been several references today to the potential of parallel human and animal research programs. And I hope that we can touch on that more in the general discussion. So I'm going to talk a little bit about some data that we've recently collected in my lab that provides some behavioral evidence that there is compensation at the behavioral level in our aged rat model. And we're in particular working with a Fisher Brown Norway hybrid rat. So they're not as diverse as the outbred Long Evans rats that Michaela Gallagher spoke of earlier, but they age with a little bit more resilience than some of the Fisher models that Carol Barnes works with. Then I'm going to talk about some evidence we have at the cellular level that may be suggestive of a compensation mechanism, but notice that there was a question mark in my title, and we'll talk a little bit about some alternative explanations for those data. And then finally, I'm going to end with some very preliminary data with some functional imaging we've done of kind of these large-scale networks that have been discussed in humans that we're doing in our rat model in an effort to kind of bridge the divide between human and animal work. So first I'm going to start with the behavioral model, and this work was done by my graduate student, Abby Hernandez, and a very talented undergrad in the lab who's gone on to medical school. And this is what we call the object-paired place association task, or we also lovingly refer to as OPA. So if you hear me say OPA, I'm not toasting, I'm referring to this behavior. And how this works is the rats just go back and forth on this two-arm radial maze. And when they're in the left arm, they do a very simple object discrimination. In this case, if they push the turtle out of the way, they get a Fruit Loop reward. If they select the baby carriage, they don't get anything. Now, when they go back to the other arm, they see the same object, but in this case now, the baby carriage is what is going to be rewarded, and the turtle's never rewarded. So they have to integrate information about where they are in space. They have to maintain some sort of working memory because they have to alternate between the two arms, and they have to integrate that with the object information in order to perform this task. And we really like this behavior because it's comparable to some work that has been done in humans that functional imaging has shown really relies on connections between the hippocampus, shown here, the prefrontal cortex, as well as the perirhinal cortex. And from inactivation lesion data that my lab has done, and also the lab of Ina Lee, we know that these same structures are involved in the OPA task, and even more importantly, that these same structures have to functionally interact to get comparable behavior on this task. And when we did this in our young and old rats, here's what we found. So what I'm showing you in this plot is the percent of animals to reach criterion. So in this case, criterion is getting over 81% correct 2 days in a row, so it's not just a fluke, plotted as a function of test day. And you can see young here represented by the white bars. The young rats start to get this behavior after about a week, and by 2 weeks, even our kind of slower learning young rats are at criterion performance. Now the old rats, they're a little bit behind. Even our best performers don't start to get it until day 9, and it takes 3 weeks for all of our old rats to reach criterion performance on this task. Another way to look at the data is to look at the number of incorrect trials that are made before the animal is considered to be at criterion performance. And that's shown here on the y-axis. And you can see that on this OPA task, the aged rats make significantly more errors before they reach criterion. And this isn't the case with kind of our control task in which they only have to do an object discrimination or a side discrimination. So they're fine on these kind of simple single item tasks, but when they have to do the more complex biconditional association, there's an aged impairment. Now, all of these animals kind of went through a larger test battery. So we tested them on this OPA task, but we also tested them on a spontaneous object recognition task. And this is something that I had worked with a lot when I was in the lab of Carol Barnes. And how this task works is you put an animal in an arena with an object that is novel to them and a familiar object, and you just look at the spontaneous exploration behavior of the animal. Now, in rodents, this is also true in babies and dogs, they'll spend more time exploring something that's novel, and you can use that as a marker for recognition memory. So in these rats, we replicated what had been seen previously by Carol Barnes's lab and others, that if you look at the percent of exploration time for the familiar object compared to the novel object, the young rats spend more time exploring the novel object, but this is not the case with the old rats. And this behavior is really dependent on the perirhinal cortex. Lesion studies have shown a number of times that if you lesion the perirhinal cortex, you see a dysfunction in this task that's comparable to what we see in the old animals. Another way to look at these data is to look at the discrimination index, which is just a bias for looking at that novel object. And you can see, again, the aged animals don't show a significant discrimination index, suggesting that these animals have an impaired perirhinal cortex. Now, why we did this cross-characterization and why this is interesting is that we have animals that have a dysfunctional perirhinal cortex that were also tested on this behavior that requires the perirhinal cortex, the hippocampus, and the prefrontal cortex. And when we looked at the performance on both tests, so what I'm showing you here is the Z-normalized performance on the spontaneous object recognition task, that's perirhinal dependent, plotted against the Z-score performance on our OPA task that requires all three structures to talk to each other. There's no correlation, which isn't much of a surprise. But what's really interesting is that in a third of our aged rats, in this nice subset, they have evidence of perirhinal cortical dysfunction, but intact performance on the OPA task. So they're performing somewhat comparable to the young animals, even in the face of perirhinal cortical dysfunction. And so now we can take these animals out and look and see what sets them apart. Is there some sort of neurobiological explanation for their better behavior on this task? So to start to get at this, we took a little bit different of an approach then. We trained animals on our OPA task, but in this case, we kind of overtrained the old rats. So we put all the animals through the tasks for 3 weeks so that our old rats were performing on par with the young. Then we had them do this OPA task for just 5 minutes. We gave them a 20-minute rest. And then we put them into a control task where they just have to alternate back and forth between the arms. Now, the reason for this kind of 5 minutes, 20 minutes control task is that when we were done with that second task, we sacrificed the animals so we could process their brain tissue for the expression of immediate early genes. And in this case, the immediate early gene ARC, which we know to be involved in learning and memory. And how this works is that after the cell fires, so this blue circle here is my schematic neuron. When it flashed yellow, that's the cell having an action potential. Within 1 to 2 minutes of that cell being active, you see 2 transcription foci for the immediate early gene ARC. So that would be the animals running around doing the OPA task, the neuron fires, you see ARC. Then you put the animal back in the home cage for that 20-minute rest. And during that rest period, the ARC RNA are actually translocating out to the cytoplasm of the cell because ARC is an effector protein that's locally translated at active synapses. And then you put the animal back to do another behavior. If the cell fires again, you will see transcription foci for the immediate early gene ARC once again, which means that you can use the subcellular location of ARC to infer the activity history of cells across the brain. So if you see ARC in cytoplasmic only, that cell fired during our OPA task. In the nucleus only, it was at the alternation task, and then if it's overlapping, it was active during both tasks. So we did this, and we looked for ARC expression in the CA1 subregion of the hippocampus, and I'm showing you those data here for the young, represented in green, and the aged rats, and which you can see overall in CA1, more cells are turned on during the OPA task relative to alternation, but there's no age difference in the activation. Now, when we looked in perirhinal cortex, again, we also saw a task effect. So more cells were active during OPA relative to alternation, but what we also saw was less activity in the perirhinal cortex, which is, again, is a replication of some work that I did when I was with Carol Barnes, that the aged perirhinal cortex isn't as activated or as excited by behavior relative to the young. Now, what we found in these data, though, that we're really excited about is that when we looked in prefrontal cortex, so in this case, we're looking in the medial prefrontal cortex, infralimbic and prelimbic, which is considered to be homologous to the human dorsolateral prefrontal cortex, we actually see that during both tests, the aged rats have an enhancement of activity in the prefrontal cortex. And this, we think, could be suggesting that there's some sort of prefrontal compensation as the perirontal cortex becomes dysfunctional. Which has also been shown with human imaging data to some extent. But the caveat here being, and this was mentioned earlier, this could also be due to a loss of neural inefficiency. And so we're looking in relation to behavior to determine if this is, in fact, compensation or inefficiency. And then the other data that I want to speak to that, again, is going to kind of hopefully bridge this gap between rodent work and human work is we're also looking at resting state functional connectivity. So with my collaborator at University of Florida, Marcelo Fibo, who's part of the National Magnet Laboratory there, he has an 11T small animal magnet that he uses. And so we took young and aged rats, and all we had done with these animals was put them on food restriction and handle them. So they were going through the procedures that they would be going through for behavioral testing, but we weren't actually doing anything with them yet other than food restriction and handling. And then we put them into the magnet, and we got resting state fMRI from them. So this is kind of the group data being shown here. We only looked at z-scores above 0.3, and each sphere corresponds to how connected that node is with the rest of the network. So larger spheres show more connections, and each line here shows specific nodes of the network that are connected. And one thing that you can kind of see just getting the gestalt of this is that at baseline, when the animals hadn't done any behavior, the young brain is more connected than the aged brain, and that had kind of been discussed earlier with human data. Then we had the animals do our OPA task. So after this initial scan, they went through OPA testing for 2 weeks. So there were still some age differences. And after they were OPA testing, you can see that actually there's a striking difference in that resting state connectivity. So that kind of resting state connectivity isn't kind of this solid architecture. It's something that's modifiable by experience. And you can see that that goes up in the young, but also very, remarkably, it goes up in the age, and to some degree, a greater extent in the aged rats compared to the young. I don't have these data yet, but we actually also took a third time point, 2 weeks later, so we can see the extent to which that enhanced connectivity maintains as they start to overlearn the task. And with that, I would like to thank the people in my lab that have really done this work, my collaborators, Dr. Jem Baizan, Ben Clark in New Mexico, Andrew Maurer, and Marcelo Fibo, of course, and then also our funding institutions. And one thing to mention before I wrap up here is Dr. Dockery mentioned this morning how one of the missions of the McKnight Brain Research Foundation is to nurture young scientists. And I thank them for that. I'm an example of that. From my graduate training through my postdoc and then starting my own lab at Florida, I have received a lot of support from the McKnight Brain Research Foundation. So thank you, and I can take questions.
Thanks very much. Questions? Great talk.
Yeah, that was a very, very nice talk. I do have just a methodological question on the fMRI part. So one of the things that you're training these rats to do is be inside of the scanner and have their brains imaged this way. And do you have a control group who doesn't receive the behavioral training but gets to scan twice?
So we don't have that in this group. Marcelo has that, and that's actually one of the reasons we took so much care to food restrict them to the weight when they were going to be doing the behavior and handle them, because he'd seen, quite large changes in connectivity just from that process alone. No, I mean that's a really good point that we should have that control group that doesn't go through scanning. I think to some extent when we have the analysis done from the third time point, we can start to get at that a little bit because they're more there's less differences between 2 and 3 versus 1 and 3 in terms of the procedure for that.
It's really potentially very interesting. It's just I'd like to see that additional control.
Yeah, no, that's an excellent point. Thank you.
Hi, Sarah. I think you know what I'm going to ask you. So, perironal cortex and hippocampus, but I would expect that your associative memory task really requires or may require postrenal or parahippocampal cortex. Do you have any sense of what role that's playing in this whole story with aging?
Yeah, so we don't have those data yet, but of course we can use that as a seed region and see how the connectivity changes between that as well. And you know, and you and I have talked about this a little bit. If it's less compromised with age and we see the perirenal connectivity go down, perirenal activity go down, with this data set, we'll really be able to look and see if there's any sort of compensation from parahippocampal postrenal. And so actually, I have a student who's going to be outlining the postrenal in rats and using that as a seed region. So yeah, we thought of you when we were talking about that as well.
Other questions?
Hi, great talk. Sort of related to that. So the OPA task is essentially a discrimination task. So I guess I'm just curious, in terms of the dentate gyrus, when you do look at arc expression, do you see any what differences do you see between the young and aged mice in terms of the OPA task? And can you make any speculations as to the computational process of pattern separation in relation to that?
Yes. So we're actually doing a lot of other work to kind of looking at, dare I say, like behavioral pattern separation, the animal's ability to discriminate as that becomes more difficult. We do have, I could say there's arc activation during the OPA task. And Carol can speak to this. Quantifying arc in the dentate gyrus is a little tricky because it's very sparse. And so you have to kind of look at the entire dentate gyrus, or at least the entire upper blade, to not bias how you're looking at that. Although one thing that has also been shown by Carol when she was working in a collaboration with Scott Small, that with aging there's less activity in the dentate gyrus. And so, I mean, it absolutely makes sense that that's a region that we should interrogate for being involved in this. The other region that we are also looking at is CA3. And Michaela Gallagher mentioned earlier that there's hyperactivity in CA3. This is seen in monkeys, in rats, and in humans. And we have a discrimination task different than this one, where we actually use Lego objects to manipulate the difficulty in the discrimination. And we do see evidence for hyperactivity in CA3 in those animals that can't do the discrimination task. So we will, you know, there's a lot of kind of nodes in the larger circuit that can be integrated into this story that we're looking at. So thank you for that question.
Other questions? Can I ask you a question, Sarah?
Of course.
Did you say that the connectivity density was maintained 3 weeks later?
So in our third time point, we don't know, we don't have that quantified yet. And so one of the questions is, as the young animals become experts at this, you would predict, although I can't confirm this yet, that the connectivity density might start to go down if they're becoming more efficient, if that's what happens as they acquire more expertise with the task, if you will. And I think that'll allow us to help address, is this enhanced activation that we see in prefrontal, is that a reflection of compensation, or is it a reflection of inefficiency?
Okay, great. Thank you for a wonderful talk. Thank you.